Welcome, and thank you for all being here today. I'm Jennifer Lippincott-Schwartz, President of the American Society for Cell Biology. I'd like to open by welcoming the leadership of the Allen Institute to the ASCB meeting. We are delighted that this major announcement about cell biology is being made here at ASCB, the world's leading international meeting of research by cell biologists. ASCB has always been the epicenter of our powerful and even more insightful science. One of the hallmarks of cell biology has been its ability to reinvent itself over and over as new technologies or new paradigms have allowed us to look more deeply into the mechanisms of life and more widely at life's vast interconnections. We have been through a whirlwind of reinventions in recent years, omics and super-resolution imaging, just to name a couple, and I predict that you are about to witness the launch of another transformation initiation in cell science. So it's my great pleasure to introduce the CEO of the Allen Institute for Brain Research, Brain Science, Alan Jones.
All right, thank you, Jennifer. Hello, and thank you all for joining us today. I'm Alan Jones. I'm the Chief Executive Officer at the Allen Institute for Brain Science. I'm joined today by a new addition to the Allen Institute team, renowned cell scientist Rick Horowitz. More than a decade ago, when he founded the Allen Institute for Brain Science, Paul Allen had the keen vision to bring together some of the best and brightest in science, with a commitment to accelerating our understanding of the human brain in both health and disease. We've delivered on that promise by creating publicly available tools used openly and freely by scientists worldwide. We know that researchers from every major academic institution, research institute, as well as biotech and pharma companies around the world use our data to advance their science, and they use it every day. Today, the family of Allen Institutes is growing. We're expanding our commitment to big, open, team-driven science by announcing a major new scientific endeavor, the Allen Institute for Cell Science. Mr. Allen is committing $100 million to investigate and model the very complex living machinery of human cells. It's a tremendous investment. Our new institute will use a goal-oriented, structured approach to systematically put the parts of the human cell together into a common framework. We will create publicly available data and models to enable scientists around the world to make predictions about cellular behavior. Cells are the battleground for every disease known to humankind. We need to better understand the inner workings of the cell in both health and disease. Before we launch into a broader discussion about specifics, we invite you to view a brief video to give you a glimpse of this incredible journey that lies ahead.
The cell, the fundamental unit of life. Each one an intricate machine, a universe unto itself. A potential target for disease.
There are things that go wrong in the genetic code, but we really haven't translated that well into what's actually going on with all of those machines. Something has changed with all of those machines that push it down a path of being diseased.
All of us begin as a single cell, growing to roughly 50 trillion by the time we're adults. That's more than 160 times the number of stars in our galaxy.
Each of our cells is comprised of a myriad of molecules, millions of them. Most of us study one molecule or two molecules at a time, maybe one machine at a time, but somehow at one point we need to put these all together.
In recent years, scientists have gained a greater understanding about the parts inside of cells, but understanding how those parts work together to produce a healthy living cell and what goes wrong when a cell is targeted by disease will take an entirely different approach.
We know that with the tools that are around today, that these are tractable problems, but they require a real concerted effort, a scaled effort to tackle them.
To take on this challenge, philanthropist Paul G. Allen has made a generous commitment to found the Allen Institute for Cell Science. The Institute will use new technologies at a large scale to study the cell as an integrated system. Big team science that can't be done in traditional academic labs.
The big aspects of it are around not only the size of the resources, but the ambitions and the goals of the project.
The goal? to create publicly available tools, data, and models that will be shared with scientists everywhere.
This includes basic scientists because alterations or mutations tell us about how the cell works, but alterations and mutations are also critical for disease.
Cancer is a great example of that, but really you can imagine just about any kind of disease, cardiovascular disease, things that affect lots of people, Alzheimer's disease. These are all diseases of cells that are going awry somehow, and we need to understand what's going on at that molecular machine level.
We live in an enormously exciting era now. New discoveries are being made monthly. And by putting together multiple cutting-edge technologies, the prospect of us making major discoveries is exceptionally high.
Accelerating our understanding of the cellular universe. The Allen Institute for Cell Science.
So now we'll talk more about the specifics of this new endeavor and how our approach at the Allen Institute for Cell Science will, in many ways, be different from what is done in the field before. The Allen Institute for Cell Science has been many years in the making. Given our success in moving the needle on brain research, Mr. Allen pushed us to explore other areas of biology that might benefit from a large-scale research effort. We worked hard to bring together experts from around the world to find these opportunities, discussing where we could make the greatest impact. With recent advances in microscopy and computer technology, we knew the time was now to launch the Institute and propel the field of cell science into a new era of discovery. Historically, cell science has been broken into many separate blocks, with individual academic labs tackling small pieces of a cell's machinery, largely in isolation. To integrate these separate research areas, we need a coordinated approach, one that creates reliably predictive models with a large multidisciplinary team focused on common goals. The Allen Institute for Cell Science will be that unifying force. We will take big team science approaches to systematically study and integrate parts of the human cell, all within one institute. We believe this kind of goal-oriented teamwork is the most effective way to achieve the greatest impact, not only for cell science, but for life science research as a whole. So to tell you a little bit more about how we plan to do that, it's my great pleasure to introduce you to the Executive Director of the Allen Institute for Cell Science, Dr. Rick Horowitz.
Thank you, Alan, and thank you all for being here today. We are embarking on an amazing journey into the exploration of the cell, the fundamental unit of life and arguably the language of biomedical research. The goal of the Allen Institute for Cell Science is to explain and predict behaviors of the cell, whether healthy, regenerating, or diseased. Over the past few decades, we've seen remarkable progress in understanding the individual pieces of cellular machinery. We've identified their organization and the structure and function of many of the molecules that comprise them. Thanks to the Human Genome Project, we now have the underlying dictionary of the cell and some of the tools needed to understand what genes determine cellular behavior. We do not, however, know how the pieces of machinery work together to produce a living cell, and how these cells interact to form living organisms. We also cannot predict cellular behaviors and how mutations, drugs, or changes in the environment influence them. Today, we have powerful technologies at our disposal to investigate and analyze cells, but the complexity of the cell requires a barrier to it. But the complexity of the cell presents a barrier to attempts to study it as an integrated system, and to do this requires a very large and multidisciplinary initiative. At the Allen Institute for Cell Science, we plan to leverage the strengths of academic research, which excels at providing insight into how individual pieces of the cell function, to create tools and models and an integrated approach that will benefit the entire cell science community. Our inaugural project, the Allen Cell Observatory, will build on the enormous recent progress in understanding the individual pieces of cellular machinery. It will take a large-scale perspective, examining and modeling the cell as a system of dynamic interacting parts. One outcome of the Cell Science Observatory will be a one-of-a-kind, novel, visual, dynamic and predictive model of the cell. This project has the potential to greatly accelerate biomedical research by providing a way to predict cellular behaviors using computer models ahead of trials, for example. Other outcomes of the Allen Cell Observatory will include a large library of gene-edited human stem cell lines, models of cellular behavior, and a library of reproducible assays and analysis tools. All of these items will be open to the public, allowing our colleagues to develop their own data analyses and models. This is a very exciting time to be a cell scientist. There is much work to be done. We look forward to leading the charge into this new era of collaboration and discovery. Thank you very much, Jennifer, Stefano, and the ASCB for hosting this announcement. Thank you.
Alan? All right, thanks, Rick, so much. I'd also like to acknowledge Congressman Chaka Fattah for attending today's announcement. We really appreciate the support. We'd now like to open up the room to questions. We've also got folks on the web, but I think we'll take questions from the room for right now. So, questions?
So we'll be starting out with stem cells, human-induced pluripotent stem cells, and we will differentiate them into one or two different cell types, perhaps cardiomyocytes and epithelial cells. So we have to take a finite set at first. Afterwards, we'll expand them and start comparing the cells and ask how different genes that dictate different cellular behaviors, how that also dictates the organization of the cell that generates these differences. But the community will probably get involved as well.
Are you going to be using a 3D growth modeling approach, or are you going to be doing it in petri dishes on surfaces as cell biologists traditionally?
So that seems to have a value judgment. So the answer is it's easier to do the imaging on a dish. The good news is that the recent advances in microscopy are allowing us to do this in 3D, so we will do it in 3D for those who are in the business, in organoids and other types of 3D structures. So the answer is yes, we will, because the differences between cells growing on a dish and cell, which we've studied for years, and cells growing in 3D is enormous. And we would like to capture that both experimentally to understand what the differences are, and because the latter is a better way of doing it.
Tell me a little bit about how this connects up and is intertwined with the neuroscience work that you've been forthcoming.
Great question. I was wondering if someone was going to ask why the first cell wasn't a neuron. And the answer is we expect these two institutes to be very collaborative, obviously. We're going to be housed in the same building at the beginning, and then actually moving into a new research building next September as well in Seattle. So the co-location will be an obvious point of interaction. We decided really, I think, very early on in the project, we wanted to not do neurons. It's very challenging to make neurons in addition, do some of the cell biology that we really want to do very systematically there. But it's our hope that once we establish the platform, that neurons will be one of those cells which will be very much next in queue to be able to start to apply this integrative approach to.
Could you, with your questions, could you go to the microphone and introduce yourself as well?
Okay.
So that we can.
I'm Sandra Mazer, and I'm a professor at the Icahn School of Medicine at Mount Sinai and chair of the Women in Cell Biology Committee. So I had two sort of interrelated questions. When you talk about the community that you expect to interact with, I was wondering about who that is and how broad it is and how are they going to connect with you. And the other thing is, as you recruit your team, you're obviously going to be going for diversity in terms of all sorts of tools. And I also was concerned that there be a diversity in the variety of peoples that you also include as your scientists.
So do you want to talk about community?
Yeah, so I can answer the second one, and the answer is yes. Of course. The first one, as far as the community goes, that's it's a community of interested people. So what we plan to do is a lot of decisions about specific aspects of the projects we're doing, because of the complexity of the cell, is going to require that we're going to have to ask people and convene them in groups, in workshops, and get their input on what we might do and how it might be done. And as the models and other things start to form, the community will similarly be engaged. So we really see a genuine engagement between the community and the work that we do, either through our outputs, which are free for them, okay, it's open access in a sense, but more importantly, in the design to a certain extent. Of course, some judgment and decisions have to be made centrally, but the cell is so complex, there is no one that I know, well, maybe one person. There's no one I know who actually could do this by themselves, and I think that the community has to be involved. So this is a deep, genuine type of an interaction.
And I would just add to that, is, from the history of the Allen Institute for Brain Science, we've had a consistent user engagement all the way back from our early days of our atlasing work, where scientists come access the data. We provide lots of tools for people to use that. So I would point to, for those of you who don't know the Allen Institute for Brain Science in this community, I would point to that example as one that we will be following. More questions from the audience?
Hi, my name is Vivian Marks. I'm with Nature. 2 questions, if I may. One is, how many people do you think you'll be hiring? Because I hear from scientists they like jobs. And the other thing is more for you. How do you get biophysicists, cell biologists, mathematicians, computer scientists to talk to one another? You talked about teams. How are you going to do that? Thank you.
So let me answer we plan to be somewhere in the vicinity of 75 people over 3+ years, so that's the rough size. The other side of it is how to get people to interact, and that's very challenging. I understand that. And in my former life, I was the director of a big NIH consortium that had engineers and mathematicians and on and on. And we have a lot of experience in that, and I can go into detail, but it's not easy. It took us a year or two to figure out how to get the cultures to start to merge. So I think the easiest non-answer is that I'm really aware of what that challenge is, and we have some experience in how to address it.
And the Allen Institute for Brain Science has been, I think, a good example of that. If you bring people in and they're all aligned to one mission, it's about bringing the people who are excited about that mission to the table, and then it's a one common mission that we're all working towards. So that's the easiest way to do it. Everybody's under one roof, and everybody is marching towards that common goal. So it's about finding the people who are most excited about being in that type of environment. We find very refreshingly that there are many people that like to work in that kind of environment. I think people are, in general, social creatures and enjoy working together. And so it attracts not necessarily people who would be better off running their own lab, but people who really have a lot of skills to offer, but aren't necessarily finding that in a traditional academic setting. Okay. Any other questions here? Steve, are we going to start taking? Okay. I'm sorry. Okay, ready for phone questions?
Signal by pressing *1 on your telephone keypad. you are using speakerphone, make sure your function is turned off to allow your signal to reach our equipment.
I'm not sure if that was a question. I'm not sure what that was. I'm sorry.
I'll give it an answer.
And we'll now take Alan Boyle with BBC News.
No, I think it was Alan Boyle of MSNBC News. That's
Oh, hello. Am I on?
Yes.
I'm sorry. So I just had a couple of quick questions. One is Paul's commitment is $100 million, but over what time period will that be executed? And I guess the other thing is that there are other cell observatories out there, and I wonder if you could tell me how the work at the Allen Institute will be different from what the other cell observatories are doing. Thanks.
Perfect question. So I will address the first question and then turn over to Rick for the compare and contrast other cell observatories. The $100 million commitment is for 5 years, so that takes us out to 2020.
So at this particular time, there are other observatories by name. There are almost no observatories in terms of what we intend to do in terms of this image-based research. There is one in Europe that's focused on the nucleus, which is one of the machines. We're going to work with them and leverage their work, but I don't know of any other observatories of any scale that's going to look at the cell in an integrated way, and deal with the images the way that we're dealing with them.
Okay.
Laboratory. In the video, you said using every cell, every disease known to humankind. I wondered if you could comment a little bit on what you mean by that and the balance between basic understanding of the cell and the sort of pathological side of things.
Well, as you know, Richard, to understand disease from a cellular point of view, you have to understand the cell. And while the basic science of the cell sounds like we know a lot, we actually don't know a lot at all. And there's an enormous amount to know. The deeper you dive, the less you find out that you know. When you look at it as a system, it hasn't been done yet. So there's an enormous amount of work to be done. No one should think that we know a lot. And as you also know, what we find out is going to have enormous ramifications on disease, because what the video was saying is that most of the mutations out there affect cellular processes. So once we have a grip on the cellular processes, then we and others can start looking at how the mutations affect the cellular behavior to know what the behaviors are that generate the problems. That's it. Yeah, I think the one point that we didn't make that I want to make is that the real key to understanding the cell is it's a dynamic spatial temporal system. Things are happening in particular places at particular times, and we need to understand that. And cancer, for example, is a case where either activities are turned on and not turned off at the right time, so the timing's wrong, or there's too many proteins expressed and they're in the wrong place. So the spatial temporal aspect feeds into many, many, many different diseases, as well as all the functioning of the machinery. So I think that's where we're going. And what's really nice about this is that it's a basic science effort with an eye to disease. So we're serious about being disease relevant, and a lot of the perturbations we'll study will be informed by the disease, but we're approaching it from, let's understand how the cell works so we can understand how that leads to disease.
So is there any more questions from the web? Okay.
We'll now take Alice Park with Time Magazine.
Great.
Hi, thank you. I just wanted to follow up specifically with regard to the stem cell library. Again, as with the observatories, there are other libraries out there. Just address what role you anticipate library at Allen will play, and whether you'll be integrating some of that information to locate.
So I'm gonna try to restate the question, Alice, because you were kind of breaking up there. I think what you were asking was to compare the stem cell libraries and the things, the engineered lines that the Allen Institute for Cell Science is going to be generating versus what's already out there and other efforts that are ongoing. Is that correct?
Yep, that's it.
Okay, cool.
I'll let you answer.
Huh?
Just answer.
Huh? Well, go ahead.
Okay. So there are many stem cell libraries. What our library will be these gene edited. That's a lot of jargon. But we're going to have hundreds of cell lines tagged with fluorescent markers so we can look at them in the cell, because as we brought up before, we have to see the cell to understand it. So these are going to be gene-edited lines. There's a few of them out there, but we're going to have a library that, as far as I know right now, does not exist.
And I want to add to that, and to tie into the question about the community before. One of the things that Rick has spent the last couple of days doing is sitting down with all of the experts in the field. We are here at the American Society for Cell Biology meeting, and there's an extraordinary amount of history and expertise to leverage about exactly where to put that fluorescent molecule so it's going to be the most effective readout. We can't do this project without all of the history and the collective wisdom that comes from this community. So we have a very systematic way in which we're going to be engaging that community. So in terms of addressing the question of how are we different from others who might be trying to do this more systematically, is really, it starts with that level of community engagement.
Governance and advisory boards, how are you going to set this up? Who is going to govern the new Cell Institute?
So the Allen Institute for Cell Science is part of the larger Allen Institute. So there's the Allen Institute for Brain Science, the Allen Institute for Cell Science, which is governed by a single governing board. There are, for each of the institutes, we have very strong scientific advisory board presence. So each of those has scientific advisory boards. And then each of the projects that we do typically have scientific advisory councils as well. So I think as part of the press materials, we announced that advisory council, some of whom are actually sitting in the audience today, for that, for the Institute for Cell Science.
You say that there's a list available currently?
Yeah, that's part of the
press release.
It's on the press release, yes.
I'll take our next question. Jennifer Cousin-Frankel with Science Magazine.
Okay. Please go ahead.
Hello, this is Jennifer Cousin-Frankel at Science. I just had a few questions. I was wondering, first, what happens after 2020? You said that initial $100 million is for 5 years. Are there certain benchmarks that you are shooting for that would then garner additional funding, or will you be subsisting largely on grants at that point? And I was also wondering whether the formation of this institute reflected any concerns that NIH is not funding enough research into cell biology at the moment, or is not funding it in the most efficient way possible.
Well, so I mean, to address the first question first, of course, more funding for cell biology from the National Institutes of Health or other places is of course welcome. For the Allen Institute for Cell Science, I like to point very specifically to the start of the Allen Institute for Brain Science, which was a 3-year project focused on the Allen Brain Atlas, which was looking at gene expression in the mouse brain. So we had a very focused project out of the gate with very defined goals, and that's very similar to what we've got here. So if you look at the Allen Institute for Brain Science today, we have 275 people. We're growing to almost 450. We've launched onto a new program in which Paul is funding the majority of the work that we do, but we are also funded by government grants and other grants as well. Okay, any more questions from the web?
And we'll now take our next question from Sandy Doughton with Seattle Times.
Great. Hi, Sandy.
Hi there. Paul Allen couldn't be here today, but he's spoken in the past about what drew him initially to brain science. But what do you know about why he was attracted to this particular problem, cell science?
Well, thanks, Sandy. I won't speak directly for Paul, but having been in many of these conversations for quite a long time, I can tell you that, and Paul has actually written, so in his own words, he recently, or I guess it was a couple years ago, I think it was an MIT Tech Review article about complexity. So the brain, as we've been learning, increasingly getting experience with how complex the challenge of the brain is, he's been looking at other areas of complexity, and biology is sort of rich with that. And so I think the cell is a logical place to start embracing some of that complexity. It's what everything else is built on. It's certainly what the brain science is ultimately built on as well. And so largely the issue of complexity is one that I've known Paul to keep coming back to.
And it shows at this time we have no further questions.
Okay. Any other final questions, or should we wrap up? Excellent. Well, thank you, everyone, for joining us today. Special thanks to the leadership of ASCB for the opportunity to announce here at the meeting. We really appreciate it. To keep up to date with everything in the months and years ahead, bookmark our website, alleninstitutecellscience.org. Thank you.